Hey, it's Mr. YS, and this is a clip on DNA replication. So, DNA replication, a replicate is like if you've got a replica piece of artwork, it's a copy. So DNA replication is DNA copying. So we're going to take the double-stranded molecule of DNA and we need to copy it. So you can see what's happening here. We've got the double helix and it's being unwound. And then each strand is being used to make a new strand. And what happens with DNA replication in eukaryotes is we start this copying at multiple points. It doesn't just start at one point and go all the way along. It starts at multiple points of replication. So these points are known as origins of initiation. And there are lots of protein complexes which are going to bind to the DNA, and including one of the enzymes that we'll look at, DNA helicase, it's going to unwind the DNA and it's going to start to copy it. So this process of DNA replication happens at multiple points. And what this does is it speeds it up, because if you start copying from all these different points, then you can do it faster. So let's imagine we're separating these two strands, and this is a bit of a review of the structure of DNA. We can imagine it as two halves. And on this half, we have, so if we draw out the letters, we've got A, then a C, then a T, then a G. On the other side, we know what we've got. We've got T, and we can work out what the other one is because of complementary base pairing. A, adenine, only fits with thymine, and cytosine only fits with guanine. So when Watson and Crick did their work to discover the structure, from the structure, the fact that they pair only in a particular way, it then suggested the mechanism of replication. And so the mechanism is you can open up the strand and then the cell can read each side. So if the enzymes involved read the A and then put in a new nucleotide, it puts in a T. And now we've got another double-stranded molecule that we can make here. And so it just, one at a time, it base pairs the correct nucleotide. So it adds in, because these things here, the whole thing which is getting added in is a nucleotide. We're only putting the letter for the nitrogenous base, but it's one at a time gets added in. And so it does the same thing with the other strand. It goes down and it reads the T, and it knows now that it needs to add in an A. And it just carries on down. And so by doing this, by opening up the strand and reading both sides and adding in the only base that can pair, it's the only one that can make the correct hydrogen bonds, then we're going to get two new molecules of DNA. We're going to end up with two double helixes. So we've now copied the DNA. But thinking about how DNA is, we need to consider what has to happen before any copying can happen. So if it was as a chromosome, it's all supercoiled, and it would only do this during mitosis. But afterwards, it could still be a bit coiled up. There could be some coiling of the DNA. And so we need to have something that relieves the strain on the DNA. So if there's any kind of coiling or any strain, we need to relieve that to unwind it, to allow things to start to copy it. So what happens is we get an enzyme which is going to come along and it will bind to the DNA, and that's DNA gyrase. It will bind and it will relieve the strain. So DNA gyrase prepares for the uncoiling that we're going to see, the separating into two strands, by relieving the strain on the DNA. So imagine really tightly wound up string. Before you could separate two strands of really tightly coiled up string, you'd have to uncoil it a bit before you could unwind it into two. And so that's our first enzyme. DNA gyrase will relieve the strain on the DNA. It will undo some of the supercoiling that might be there. So here we have a section of DNA where ahead of it, the protein that we just talked about, gyrase, would be ahead here. And so we don't usually label gyrase on any of these DNA replication diagrams. But we can see that there's something else there. We've got another protein which is unwinding the DNA double helix. It's separated it into two strands. So it's unwound it, and now we've got this separation between them. So how could it have done this? What was holding together the DNA? Well, holding together the DNA down the backbone is covalent bonding, but in between was hydrogen bonds. So this protein is called helicase because it was to do with the idea of unwinding the helix. So helicase unwinds the DNA double helix, and it does this by breaking the hydrogen bonding. So it breaks the hydrogen bonding and unwinds the DNA into two strands. And now the code that's along there, whatever the code is, can be read on both sides. Now, if we were thinking about what this is, we've got a strand where it's double-stranded and then it's split into two. So in diagrams, this part here is called a replication fork. So we would say at the replication fork. So this fork is created by the helicase. So helicase, by unwinding and separating the two strands, has created a replication fork. And then after the replication fork, it goes back together. So what we've seen so far is helicase unwinds the DNA by breaking hydrogen bonds, and it separates the two strands to create a replication fork. And now what we're going to do is use each strand of the parent DNA, so that's the original DNA, as a template for synthesis. So we're going to use the existing DNA to know what new bases, new nucleotides to add in. So this is called, DNA replication is called semi-conservative. Why is it called that? Well, in each, if we've got this as our original DNA, if we open it up and then we add in a new strand of DNA using the original, using the parental, so if that's the parental DNA, we can see that afterwards in our two molecules of DNA that we've created, we've gone from one molecule to two molecules of DNA over here. In each of them, we have half parental DNA and half new DNA. And so we can think of semi meaning half conservative. To conserve is to keep. So we've kept half the parental DNA in the new strand, and we have half newly made DNA. And that's going to be made from nucleotides, which are, they're just available, they're floating around. So these are new nucleotides, and they're floating around in the nucleus. next we can see that we have a new enzyme which is coming to bind. So we had over here, we have helicase, which has unwound the DNA double helix. Now something that you might see, but you don't really need it for the marks, is these proteins. These are SSBs, single strand binding proteins. And what they do is they stop it from zipping up again. So they're stopping the DNA from becoming a double-stranded molecule, because these want to bond together through hydrogen bonding. These two, all the bases along through complementary base pairing, they want to go back together. So these proteins stop it from zipping back up. But that's a really high-level point. All right, so we've got primase, and you can see it's binding to the DNA on one of the strands. And the reason it needs to do that is that our building enzyme, we've got an enzyme that's going to build the new strand. It can only join to a double-stranded molecule. So if we've got a single strand along the top, it can't bind. So we need to have at least a small section where we have a double strand, and that's going to allow then our next enzyme to come in and bind. So primase, that we could call DNA primase, it's going to add in that section there some new nucleotides, and these are RNA. So they're not DNA, they're RNA, but what they do is they make a short section that is double-stranded. So you can think of it like a roller coaster. A roller coaster can only go down on 2 tracks. And so to attach, we need to have both tracks there. So primase comes in and it adds a short section of RNA nucleotides. And we call that short section, it's the primer, because it primes the DNA for being copied. So what we've seen is an RNA primer is synthesized, is made on the parental DNA, because both of those strands are parental DNA, using primase. And this is going to happen on what we call both the leading and the lagging strands, but we'll get to that next. So here we see now we've got the RNA primer, and now we've got a double-stranded section which is going to allow our building enzyme to come and bind. And this allows the attachment of our builder. So this is going to build the new train track. This is DNA polymerase, and it is number 3, DNA polymerase 3. So what you couldn't quite see is that underneath that, what it's binding to, it's binding at the primer. So DNA polymerase 3 binds at the primer, which is allowing it to attach. Then DNA polymerase 3 is moving along and it's reading. So as it reads the bases, it can now put in the correct matching base. And these bases, these nucleotides, so we've got A's, we've got T's, we've got G's, we've got C's, and they're all just floating around. They float around, and because this is an enzyme, when the correct nucleotide hits the active site, it will be incorporated and now will form these new bonds. So through hydrogen bonding between the correct nucleotide and the parental strand, through complementary base pairing, we're going to create a new strand. So it's just going to keep moving along in this direction. And you can see that it's going in the direction we've got a 5' end up there, it's got a 3' end there. So it moves from 5' to 3'. And it's adding in new nucleotides according to complementary base pairing. Now let's just review the structure of DNA to understand what we mean by adding nucleotides in a 5' to 3' direction. So here's our double-stranded molecule. And we label these carbons, these are all carbons in the deoxyribose sugar, we label those with numbers 1, 2, 3, 4, and at the top, although we don't draw it out, at the top there is 5. So if we go down to the bottom one, 1, 2, 3, 4, 5. And we can see that if we look at the top is 5, so we call that end 5', and the bottom is 3, so that's 3'. So if we look down in that direction, so going top to bottom, it is a 5' to 3' direction. So DNA polymerase 3 can attach, so it'd be attaching, this would be the parental DNA that it would be reading, and it would be adding nucleotides at this end. So the new nucleotide would have to be added on at the 3' end. So what it does as an enzyme is it adds in a new nucleotide by creating a new covalent bond. So it's got an enzymatic function. It's adding in, making the phosphodiester bond. It's creating the new bond for the sugar phosphate backbone. So what it does is it reads this side. If it was reading a C here, the only thing that could come into the active site and fit would be a G, and it would put the G in at the 3' end, linking it and creating this bond. So it would create that bond to add in the G over here. Now, most of the diagrams, they don't label it that way, they label it sideways. So let's see if we can work out which end is a 5' and which end is a 3' if we're thinking about moving in this direction. Let's go around and label them. Well, we label the right-hand side, so this is 1, 2, 3, 4, 5. So that's 5', and therefore this is 3'. And then we know it's going to be the opposite because it's anti-parallel. 1, 2, 3, 4, 5. That's the 5' end. And down to 1, 2, 3. That's the 3' end. So if we're going from left to right, that side is 5' to 3', but the other is going to be 3' to 5' because they're anti-parallel. Okay, so if we remember that, we need to remember that DNA polymerase 3 can only work in one direction. It can only add a new nucleotide if the top was the parental DNA and this was the new strand at the bottom. It could only add a new nucleotide in down here. It could only add it at the 3' end. So an easy way to remember that is that if this is the house, kind of looks like a house, it can only add in at the bottom. It adds in at the bottom of the house, not at the top. Let's have a look at this on this diagram. So sometimes you'll have to kind of follow the strand along. So it's telling us that the red, that's 5' there. So if we follow the strand along, the other end of the red should be 3'. And if we follow the blue, which was 5' there, we follow the blue round, this end must be 3'. So we know that they're anti-parallel. If this top strand, the top parental, is running 3' to 5', then the new strand will be running the opposite. It will be running 5' to 3'. So that's fine. This enzyme, DNA polymerase 3, can keep adding in towards our fork in the road. It can keep going, and as fast as this unzips, as fast as the helicase unzips, this will move towards adding in new nucleotides. So that's fine. But if we think about what's happening on the bottom strand, let's follow it round. This end here, we said that was the 3' end, and that means that this is towards 5'. Now that means that the other strand, the new strand which is going to be made, is the opposite of that. The new strand must be 3' here and 5' up there, because it's the opposite. That means that we can't just get another polymerase attaching and adding towards the zip and continuously synthesizing towards the fork. That would be too easy, too convenient. Really what it is that this will have evolved to work in one direction, 5' to 3'. And so it was easier in terms of evolution to figure out a way to get it to copy the other strand than to make a whole new enzyme for it. So we can see here, if this is 3' to 5', but we said our polymerase can only work 5' to 3', it can only go in the other direction. So it's only going to be able to add new nucleotides that way. So it could bind, add some new nucleotides for a section, but then more will be unzipped. And what this leads to is the idea of a strand that we call the leading strand, where adding in 5' to 3', we can go towards the fork. So it's going towards the fork on that strand, but 5' to 3' on the other one is going away from the zip. And that means that we can open up, so as we unzip, we can do a section, but that's it. It can only be done in sections. And then more will have been unzipped. More DNA up here. This is newly unzipped. And the newly unzipped DNA now needs to be copied as well. And so what we're going to have is this idea of it doing a little section at a time, and that's all it can do. Here's our summary then. So we had DNA polymerase 3, the builder. It copies DNA in a 5' to 3' direction, and it does that on both the leading and the lagging strand. DNA polymerase 3 starts adding new nucleotides at the primer, and this has been a phrase that they're really keen on in the IB mark schemes. So at the primer, according to complementary base pairing. Adenine pairs with thymine, cytosine pairs with guanine. You are not allowed to say A and T, G and C. The other thing that it does, so this is a high-level statement, is that it also checks, it proofreads, it checks for errors. So as it adds in the nucleotide, it checks that it's the right nucleotide. And because of the DNA polymerase only being able to go one direction, synthesis is continuous on the leading strand because it can go towards the zip, but synthesis is discontinuous on the lagging strand. It has to be done in small sections. And these sections are called, so for each section where we've unwound and we can do that section away from the fork, these are called Okazaki fragments. let's have a look at what's happening on the lagging strand. So the leading strand was easy. We added one primer, and then it's just going to keep adding in the DNA polymerase 3. We'll keep adding in new nucleotides towards the zip, towards the replication fork. But for the lagging strand, it also is going to need a primer. So this is one section, this is already done. So this section's already been done, so we can kind of ignore that. Let's ignore this section here. But you can see what's needed. We need, as well, on the lagging strand, both leading and lagging strand, we need a primer. So we've got primase coming in here. Primase is coming in, and it's going to add that short section of RNA. So it adds in the RNA primer to make it double-stranded so that DNA polymerase 3 can attach. So we can see now we've got the primer. We can have DNA, because this is the same thing, DNA polymerase 3. So polymerase 3 is going to bind, and if it's adding 5' to 3', this is the 3' end, which means the other strand is 3' to 5'. So it's going to add, but you can see it can only add a bit, because then it's going to reach a point where either it was a already done section, or if it's the very starting bit, there's no, it can't keep going because the DNA is not unwound. And so once it does that little section now, ahead of it, you can see there's already a new section of DNA that's been unzipped. And now it's going to have to come and bind here and run along to the point where it reaches the double strand, and then it will fall off. So it's going to be done in these little sections, and those sections are Okazaki fragments. Now there's also a problem. So this is RNA. So on the lagging strand, we've got a lot of problems to solve. This is an RNA section of nucleotides, and we don't want RNA. So this needs to be replaced by DNA. And so we need an enzyme to do that. Here we see that our polymerase 3 has run along until it gets to that RNA primer at the start of the section that's already been done. So it gets to that point, and then it's going to release from the DNA. So this has been released, and underneath this was the RNA section. So now we see our new enzyme has come in. So this is going to make a small bit of DNA. Because it's making DNA, it is still a DNA polymerase. Because that's the idea of making new DNA. But this is DNA polymerase 1. So this is a different enzyme. DNA polymerase 1 comes in, and what it's going to do is replace that section of RNA with a section of DNA. So it replaces the RNA primer with DNA. So here's our DNA polymerase 1. That we just use the Roman numeral I, and it's replaced you can see here there's no RNA anymore. So it's replaced the RNA with DNA, and that's good, but there's still a problem. Here we can see a gap. There's a gap where there is no bond between those nucleotides. So there's a gap, or we could say it's a nick in the DNA. And that needs to be sealed. So here we have our final enzyme that's going to come in, we have DNA ligase. DNA ligase is going to come in, and the gap that was there between, so it's, if we're imagining like this train track, on one side there was a gap in the train track, and that gap is really between, it was the sugar phosphate backbone. So it means really what it's doing, DNA ligase, is it's making a new sugar phosphate. It's joining it up. That's a covalent bond between the sugar and the phosphate group along the DNA backbone. So we can say that DNA ligase joins the fragments, joins the Okazaki fragments, by forming covalent sugar phosphate bonds. It's come along and it's sealed those gaps. So here's our rather long summary. Now this is usually an 8-mark question, and so you could miss quite a lot of the points and still get the marks. So we could talk about when it happens. DNA replication is during S phase of interphase, before mitosis. We saw that gyrase prepares for the uncoiling that helicase is going to do by relieving the strain on the DNA, relieving the supercoiling. Then we had our first enzyme on our replication fork, which was helicase, which was unwinding the double helix, separating the strands by breaking the hydrogen bonds between the two strands. Then each strand of the parent DNA is a template for making new DNA. And so we talked about the idea that if we've got 2 parental strands and we add in a new strand for each, then what we get in the final is we get 1 strand, which was the original parental, and 1 strand, which was new. And so this was semi-conservative replication. In order for DNA polymerase 3 to bind, we had to have an RNA primer to make it double-stranded. So primase, or DNA primase, makes a short RNA primer on both the leading and the lagging strands, allowing our DNA polymerase 3, our building enzyme, to bind at the primer. DNA polymerase 3 then copied in a 5' to 3' direction on both the leading and the lagging strands. And it does that by adding in new nucleotides at the primer, at the 3' end, according to complementary base pairing, where adenine would pair with thymine and cytosine would pair with guanine. It could also check for errors. Now, one thing to mention here, which is a really high-level thing, is that the new nucleotides are nucleotide triphosphates. So a nucleotide, when it's added into DNA, is a phosphate, so it's only 1 phosphate linked to the next nucleotide, the deoxyribose sugar, and the nitrogenous base. But what's used to add in is a nucleotide triphosphate, which has got another 2 phosphates on here. And the reason is that as they're added in by the hydrolysis of this and the new bonds it forms with water, the removal of these 2 phosphate groups, it provides the energy for DNA replication. So it's a bit like ATP, because ATP was adenosine triphosphate, where the removal of one of those phosphates led to the release of energy. And that's, what's produced from respiration. We produce ATP, our form of energy. And so the nucleotide triphosphates are a form of energy for powering DNA replication. Now that's a very high level, 7-level statement, but you might see that somewhere and wonder what it is. Okay. So the synthesis on the leading strand is continuous because it can run 5' to 3' towards the zip, towards the replication fork. But the synthesis on the lagging strand is discontinuous because it can't run towards the replication fork 5' to 3'. It has to run away 5' to 3', leading to the formation of Okazaki fragments. We also had the need to replace, and this happens both on the leading and the lagging strand, polymerase 3 needs to replace the RNA primer with DNA, and ligase needs to join Okazaki fragments, and it also joins on the leading strand as well. It needs to join the gap in the double helix, which was caused by the RNA primer being there to start.